Hello and welcome to Botany 101. I'm Angela Nishimoto. Okay, so today we're going to cover a new topic called genetics. Okay, and this outline of topics is going to be quite a heavy one, right? We're hopefully going to cover DNA, DNA replication, protein synthesis, mutations, translocations and inversions, two types of mutations. We're going to ploidy again. Remember we talked about ploidy last time? We're going to talk about Gregor Mendel and his experiments, monohybrid crosses, dihybrid cross, genetic terms, and we're going to cover genetic problems because your learning log 2 will be assigned during the course of today's lecture. Okay, Barbara McClintock caused major shifts in the way we view gene organizations. Okay, she was a geneticist, a corn geneticist at Cold Spring Harbor and in the 1940s and the 1950s. Okay, what she noticed in her corn were streaks of color where they didn't belong. Okay, there are patches of yellow and green leaves, patches of green and white leaves, and spots of color in corn kernels that should have been colorless. Okay, these were the result of transposition, which is the movement of a piece of a chromosome to another location on the same chromosome or on a different chromosome. Okay, and these transposable elements are called also transposons or jumping genes in corn. Okay, so the yellow and the green leaf areas, okay, were because transposons were inserted into a gene that was used to do with chlorophyll synthesis. Okay, the green areas on the white leaves resulted when transposons moved out of a chlorophyll synthesis gene, causing it to function again. Okay, she got the Nobel Prize in 1983. Okay. Now 85%, amazingly, 85% of the DNA in corn actually comes from these transposons, okay? And transposons are capable of causing reversible mutations, okay, as we saw in the green areas on the white leaves, okay? Now James Watson and Francis Crick, okay, delineated the structure of DNA in 1953, and they kind of ushered in a new field of molecular genetics. Okay, so if you recall, chromosomes in the nucleus has genetic information that is passed on from one generation to the next. Okay, so we're going to go into molecular genetics first. Okay, so chromosomes are made of DNA and protein, recall, yes? And the molecule of genetics is DNA, okay? This is review. And DNA is deoxyribonucleic acid, okay? And this DNA, the subunits are called nucleotides, right? Remember that? Okay, so nucleotides are made up of a 5-carbon sugar. In this case, it's called deoxyribose. Okay, it's also made up of phosphate group, the nucleotide is. Okay, and thirdly, you have a nitrogenous base. Okay, the nitrogenous bases, there are 2 purines, which have a double ring structure called adenine and guanine, abbreviated A and G. And then you have pyrimidines that have a single ring structure. Okay, they're called cytosine and thymine. Okay, all right. So you have double ring structures in the purines, single ring structure in the pyrimidines. Okay, so the structure of DNA is like a ladder that's twisted into a double helix. You have alternating sugar and phosphate groups on the edges of the molecule. Okay, and each sugar is also bound to a base. Okay, so A, T, and C and G, adenine, thymine, cytosine, and guanine, these are all those nitrogenous bases, right? Okay, so what these bases will do is they will hydrogen bond from one side of the molecule to the other side, the other strand. Okay, A, double hydrogen bonding with T, okay, and C, hydrogen bonding with G, okay, a triple hydrogen bond between C and G, okay. So you have the sugar phosphate backbone, okay, and the bases are hydrogen bonded. Now Watson and Crick, they actually put together the structure of DNA using information from other people. Okay, for one thing, there was Linus Pauling, okay, of Caltech. Okay, he hypothesized that the DNA might actually be similar in structure to protein structure. Okay, and Linus Pauling was the discoverer of the alpha helix, which is shown here, okay, which is a second level of protein structure in proteins, okay, where the polypeptide actually forms a helical structure. Okay, so he thought that maybe DNA might also be some kind of helix. He thought it was like the alpha helix, though. Okay. Now Rosalind Franklin did work in X-ray crystallography. Okay, and what X-ray crystallography does, okay, it actually takes a molecule, okay, the person who is doing this X-ray crystallography, they take a molecule like DNA, and using a very painstaking process, they crystallize it. Then what they do is after that, they have a crystal, they bombard the crystal with X-rays, and that creates a refraction pattern, which is what you see here on this computer screen, right here. Okay, so right here is an X-ray crystallograph. Okay, so from the work that she did, it showed that DNA is made up of repeating units and had a helical structure, okay, and was also symmetrical all the way up and down the molecule. Okay, now Erwin Chargraff, okay, kind of delineated Chargraff's rules. Chargraff's rule, which is that the number of purines, remember, G and A, or guanine and adenine, the number of purines equals the number of pyrimidines, or cytosine and thymine. Okay, so you have the double ring structures and you have the single ring structures. Okay, so the number of purines equals the number of pyrimidines. Okay, so Watson and Crick actually put together this information from these different sources, and they also built chemical models. Okay, people laughed at them because they thought that generally introductory students in chemistry were the only ones that actually built chemical models. Okay, so there are people. So there was Watson and Crick in their lab building these large models, and then people sort of hooting at them and sort of saying that they're wasting their time. Okay. Anyway, so they were able to actually show that adenine paired with thymine in a double hydrogen bond and cytosine paired with guanine in a triple hydrogen bond across the two rungs of the ladder, okay, which was a sugar phosphate backbone. Okay, so they were able to actually show that this is how it was, that a two-ring structure paired with a three-ring structure. Okay, a two-ring structure paired with a one-ring structure. Okay, because the molecule was symmetrical all the way up and down its length. Okay, so they were able to infer the structure of DNA from other evidence from other people. Okay, now DNA functions. What does it do? Okay, among other things, it stores genetic information. Okay, and most of this genetic information is in the nucleus of the cell, compartmentalized. Okay, and what it is it has information in the sequence of nucleotides, A and T and C and G. So the sequence of nucleotides making up DNA actually code for genes, okay? Or they're actually genes that code for proteins. Okay, so GAATCC would code for amino acids that would be different than CTTAGA. Okay? So before they actually figured out the structure of DNA and actually broke the genetic code, before they thought that DNA was much too simple to have so much information that was used for the survival of every living thing on the planet. Okay? But plants have millions or billions of base pairs. Okay? So anyway, so genes actually control physical traits. How? It's because a gene segment encodes proteins, okay? So proteins are stored, or proteins are used as structures, okay? Or they're used as enzymes. And every activity in the cell happens and is controlled by enzymes, okay? Now the genome is all the DNA in an organism's chromosomes. Okay, all the organisms' chromosomes all make up the genome. Okay, now in a complex eukaryote, there are 10,000 to 50,000 genes. Okay, remember that each gene encodes a protein. Okay, so in terms of genes of plants that are not really closely related to bacteria that much. So bacteria and plants are not that closely related, even though they are related because every living thing on the planet is related to each other. Okay, but the bacteria versus plants, there are many differences in the genes that they have. Okay, whereas plants, such as closely related, such as corn and teosinte, they're similar. Okay, they have similar genes, yet they're very different looking. So the little bit of difference in the genes between corn and teosinte allows them to be very different in appearance, okay, as what they physically look like, their phenotype. Okay. Now DNA replication, when does it happen? Do you remember when it happens? Okay, think about it. Okay, I'm not going to give you the answer because we went over this maybe 2 or 3 times already, so I'm going to leave it to you to answer that question. So when does DNA replication happen? Okay, so Watson and Crick actually predicted that the DNA would be unzipped, you know, where the hydrogen bonding happened. The hydrogen bonding will be broken by an enzyme, okay, and that enzyme actually turned out to be DNA polymerase. He named it DNA polymerase, okay. Okay, so each strand, remember that DNA is double-stranded, so each strand can serve as a template for a new strand. Okay, that makes complete sense, because if you know the sequence of bases on one strand, you would know the sequence of bases on another strand, because A always pairs with T and C always pairs with G. Okay. Okay, so the new strand has all the information to build a new strand on it. Okay, that's what they call semi-conservative replication. Semi-conservative because half of the original DNA molecule is conserved to make a new strand. Okay, so each of the 2 new DNA molecules has an old strand and a new strand. That's why they call it semi-conservative, half conservative. Okay, now the nucleus has enzymes that detect and correct mistakes, okay, that proofread, okay, the new DNA, okay, they'll correct mistakes such as G pairing with A or T pairing with C, okay, but still, about 1 in 1 million bases are incorrect when DNA is replicated, okay, 1 in a million, okay. Now each corn cell has about 4 billion base pairs, okay, in the nucleus, so there are approximately 4,000 mutations that happen every time the cells divide. Okay, every time a cell divides, there are 4,000 mistakes or 4,000 mutations in a corn cell. Okay. Now genes also do gene expression. Okay. So each cell has the same information. Okay, so a cell in the root has all the same genes as a cell in a stem, okay, as the cells in a flower, okay? But different subsets are read, okay? All right? So different subsets of the information are read. Not all the information is expressed in every cell, okay? So an epidermal cell of a leaf, okay, secretes a waxy cuticle, right? So the epidermal cells express genes that encode enzymes that are needed to build the cuticle, okay? Whereas in another place in the leaf, in the mesophyll cell, they do chlorophyll synthesis, okay? They're highly specialized for photosynthesis, right? So what they'll do is they'll express a sets of genes that encode enzymes for the synthesis of chlorophyll, okay? So one of the most active areas of research in biology has to do with how organisms actually express their genes, okay? Now, transcription and translation, that leads to protein synthesis. Okay, remember the enzymes control everything that happens inside of the cell. Okay, so there's another kind of nucleic acid that is involved in protein synthesis. It's known as RNA, or ribonucleic acid. Okay, so some differences from DNA that RNA has. Okay, ribonucleic acid. Okay, RNA is usually a shorter or smaller molecule than DNA. RNA is single-stranded. If you recall, DNA is double-stranded. Okay, RNA has a different sugar. It has a ribose sugar, whereas DNA has a deoxyribose sugar. Okay, and RNA has bases of adenine pairing with uracil, okay, AU, okay, and cytosine and guanine, okay, C and G, okay. So A and U pair, C and G pair. If you recall, in DNA, A and T pair and C and G pair, okay. So uracil replaces the thymine base, okay. So first of all, we have messenger RNA. So there are 3 different types of RNAs. You have messenger RNA, also known as mRNA. Okay, the message in mRNA is translated to produce proteins. So the messenger RNA carries a genetic message, okay, that is transcribed from DNA. Okay, now transfer RNA. So here's a messenger RNA. Okay, here's a bases that actually make up this particular strand of messenger RNA. And we also have transfer RNA or tRNA. Now tRNA is also single-stranded, but what they'll do is that the tRNAs will tend to hydrogen bond with other parts of itself, forming kind of like an interesting looking sort of molecule, okay? On one side of the molecule, you have the amino acid binding sites, okay? Because the transfer RNA will actually help translate, the translator, okay? So it has an amino acid binding site on one side, and it has something called the anticodon on the other side. We'll talk more about that later, okay? Now, ribosomal RNA or rRNA is a component of the ribosome. Remember that ribosomes are places where proteins are constructed. They're actually like anvils upon which proteins are brought together, or actually put together. Okay? Now, transcription happens in the nucleus. Okay? Transcription happens in the nucleus because it involves DNA, and the DNA in the form of chromosomes, okay, never leaves the nucleus. Okay? They're actually sequestered in the nucleus for its own protection. They're like the master blueprint, right? So this is aided by RNA polymerase. This is another enzyme, and this is actually used to make RNA. Okay, first of all, the RNA polymerase attaches at an area called the promoter region. Okay, the promoter region is a sequence on DNA that acts as a flag to signal that a gene is ahead. Okay, it causes the enzyme to associate with one strand of the DNA and begin copying the gene. Okay. Then RNA polymerase adds RNA nucleotides, okay, until it reads the terminator region, okay. Then the enzymes fall off DNA, okay. Then the RNA transcript is made that way, okay, that's called messenger RNA, okay, or mRNA. Okay, so we have a image coming up here to kind of illustrate the concepts. Okay, so this large blue thing is RNA polymerase, which stands for the enzyme RNA polymerase. Here you have the strand of DNA. So RNA polymerase helps to unwind the DNA at one end. Okay, and then when it unwinds it, you know, the RNA actually comes onto a promoter region that actually tells them when to start doing transcription. Okay, so it's making a RNA transcript from a DNA template. Okay, just like just like you know what the other strand of DNA is, if you know what one strand is, you'll also know what RNA is. Okay, made from a DNA template. All right. Okay. Because if there's an A here, then you know that the corresponding nucleotide on the RNA has to be U, okay? If it's actually T on the DNA, you know that the corresponding complementary nucleotide is going to be an A, okay? If it's a C, you know it's going to be a G, and if it's a G, you know it's going to be a C, okay? So here's in red, the mRNA strand, okay, so here you have the nucleotides coming in, joined, okay, to each other in covalent bonding along the sugar phosphate backbone, okay, by RNA polymerase, okay, and then at the other end, after transcription has finished on this part, the DNA strand is rewound, okay. It's a very complicated, very fascinating sort of process that is transcription. Okay. Now translation. Okay, so transcription actually allows for the transcription of mRNA from a DNA template. Right now, translation happens in the cytoplasm. So what happens is that the mRNA is processed, or the RNA is processed, becomes mRNA, okay? Then it leaves the nucleus and goes into the cytoplasm. Now the genetic code is read in triplets of nucleotides, okay, on the mRNA transcript, okay? And these triplets of nucleotides are called codons, okay? So the order of codons, so the read in triplets of bases called codons, okay? So the order of codons is equivalent to the order of amino acids, okay? Say for example, AUG actually codes for the amino acid methionine, okay? But AGU codes for the amino acid serine, okay? Another series of triplets of bases, another codon, AGC, also codes for serine. Okay, but AUG will always code for methionine, AGU and AGC will always code for serine. That's why they say that the genetic code is redundant, but it's not ambiguous. Furthermore, the genetic code is universal. Okay, the same code is used in bacteria, in protistans, in fungi, in animals, and in plants. So except with very few exceptions, the genetic code is universal, okay? Now the DNA translator or decoder, what it does, it has the anticodon. We talked about that a little bit before, and I said we talk about it later. Now we're talking about it now. The anticodon is on one side. It binds to the complementary mRNA codon by base pairing, okay? And on the other side is the amino acid binding site with the correct amino acid, okay? So here's showing both transcription and translation. Okay, here's DNA, okay, coding for RNA, and this messenger RNA actually is fastened on by the ribosome, the large subunit and the small subunit of the ribosome. Okay, and we'll go into the details in a little bit, but here you see the polypeptide forming. Okay, polypeptides forming proteins. Okay. Okay, first of all, you start tRNA with methionine, remember AUG, yeah? AUG will be the start codon on the messenger RNA and the anticodon on the transfer RNA, okay? So the UAC anticodon will pair with the AUG codon. That makes sense, right? Since U pairs with A pairs with U, and C pairs with G. Okay, so the UAC anticodon pairs with the AUG codon and joins the ribosome. So this is sort of what it looks like when translation is going on. Okay, you have the large subunit, you have the small subunit. Okay, you have the E site, the P site, and the A site, in which the transfer RNAs actually dock. Okay. Okay, then the next tRNA comes in and pairs with the next codon. Okay, the anticodon pairs with the next codon. Okay. And attaches the amino acid to the other amino acid in a peptide bond. That's what you see here. Okay, so you have this first tRNA matching up with the messenger RNA codon, the anticodon matching up with the codon. Another tRNA coming in, matching up its anticodon with the codon, and then the amino acids joining each other, forming a growing polypeptide chain. Okay, then the complex moves over, the tRNA leaves, and another tRNA comes in, and the process continues. So it's a ribosome that's actually moving along the messenger RNA. Okay, sort of the opposite. If you know what a typewriter is, a lot of people don't know what a typewriter is. It's a thing that actually people would type at to produce documents, which people use computers for nowadays. Anyway, so I had a job as a secretary for a few times, you know. So what typewriter does is actually has a typewriter ribbon in it that moves. Okay, if you think of the ribbon as being the messenger RNA and the ribosome being the typewriter, it's like the typewriter is moving along the ribbon. Okay. Okay, so this process continues until a stop codon is reached. Okay, the 3 stop codons do not code for any amino acids. Okay, and you need to know these, just like you need to know that the start codon is AUG. Okay. That codes for methionine, right? So UAA, UAG, and UGA, those are the 3 stop codons. They do not code for amino acids. They just actually tell the different machineries when to stop doing translation. Okay. Okay, so here you go. Here's that same structure again. Here's a large subunit of the ribosome, the small subunit of the ribosome, the mRNA, okay, and the transfer RNAs and the sites, the binding sites here, okay, the anticodon and the codon, okay, the amino acids represented by these little balls. But remember that an amino acid is made up of a central carbon, carboxyl group, an amino group, a hydrogen, and also an R group, right? So each amino acid is actually the same, except the R groups are different. Remember that? Okay. Now the flow of genetic information, okay, goes from DNA to RNA to protein. Okay, so the DNA in the nucleus, okay, acts as a template for the transcription of messenger RNA. Okay, the messenger RNA is taken to the cytoplasm, okay, where the mRNA is then translated into a polypeptide, which forms a protein. Okay. Now, mutation are changes in DNA sequence, okay, changes in the nucleotide sequence, okay. It happens all the time, like we talked about with our corn example a little while ago, right? So it happens every time DNA replicates, you have mutation, okay. And mutagens, there are these substances and situations called mutagens, also cause mutations, okay, such as UV light, ionizing radiation, and chemicals. In fact, Thomas Hunt Morgan, who became very famous as a person who did work using fruit flies, genetic work, okay, so he actually used X-rays. He bombarded fruit fly larvae and eggs with X-rays, actually cause mutations like legs coming out of the forehead of the insect instead of antenna. Okay. Now, DNA repair oftentimes corrects mistakes. Okay. And if it's uncorrected, the mutation occurs. Now, most mutations are silent mutations with no visible consequences. Okay. And most DNA in the cell is non-coding. Okay, so even change coding sequences may produce proteins that can still function. Okay, so mutations can be either somatic mutations or germline mutations. Okay, somatic mutations happen in body cells. Okay, so they will exist in all cells that are produced by the mutant cell. Okay, it can result in a sport. Okay, a sport is a branch on a plant that looks different from others on the same plant. Okay, a sport is actually a mutated branch, okay, that has mutated cells. So navel oranges and red delicious apples, apples and oranges, okay. Navel oranges and red delicious apples actually originated from sports, okay. Now we have germline mutations, and these happen in cells that produce gametes, okay, and they're passed on through future generations through the seeds, okay. So these germline mutations are not usually noticed until it's passed on to the daughter plants, but they've been very important for the development of horticultural and agronomical varieties of plants, okay. So mutation can lead to variability in all traits, okay? So all traits, all genetic variability in nature comes about by mutation, okay? So though we tend to think of mutations as bad, they are an important feature of DNA. They're responsible for different flower colors and smells. They're different for different fruit tastes and textures. They're different for grain, they're responsible for grain yield and quality. And they're also important for disease and stress tolerances, okay? So the accumulation of mutation leads to all the genetic variability that happens on our planet, okay? So all genetic variability in nature comes about by mutation, okay? And mutation, accumulation of mutation is a mechanism that drives evolution. Okay, so we're going to be talking about evolution in an upcoming lecture. All right? Now, during the mid-1900s, okay, corn cytogenetics, okay, dominated biology due to Barbara McClintock and other corn geneticists. Okay, so cytogenetics, what is it? Okay, it's a study of chromosome behavior and structure. Okay, so this is a karyotype, of course, showing the chromosomes, I think, of a human being. It looks like there's 46 chromosomes. Here's X and Y, so this is the karyotype of a human male. Okay, so cytogenetics is a study of chromosome behavior and structure. There's a X chromosome, there's a Y chromosome, yeah? Chromosome 1, chromosome 2, etc. Okay, up to chromosome 22, sorry. Okay, anyway, so the study of chromosome behavior and structure, okay, from a genetic point of view. Okay, so sometimes a chromosome will break. Okay, so you have changes in chromosome structure. Sometimes a chromosome will break. You know, it's a physical structure, and like other physical structures, it can break. Okay, so the damage can be repaired sometimes by enzymes, but sometimes a broken chromosome becomes reorganized. Okay, the most important reorganizations of chromosomes include inversions and translocations. Okay, so inversions. Okay, inversions, what happens is that the chromosome is broken and reinserted in an opposite orientation. Okay, so a chromosome that reads A, B, C, D after an inversion event may read A, B, D, C, E. Okay, so the inverted chromosome can pair up with its homologue, but crossing over will oftentimes make gametes that have some duplicated ones on the chromosome or are missing segments of the chromosome. So therefore, oftentimes the gametes resulting from inversions are less fertile than normal. Okay. Now we also have translocations, and here's a good illustration of a translocation. Okay, it's when a piece of chromosomes are broken and attached to another chromosome. Okay, so here's looking at, so those are the same as transposons, right? It's translocations. So here you have two different chromosomes, chromosome 4 and chromosome 20. Okay, so this one breaks here and this one breaks here, and they actually exchange segments. Okay, so what you have is reattaching to a different chromosome. So this is what you call a translocation. Okay. Now, generally translocations, okay, because gametes have unusual combinations of chromosomes, they are also less fertile than normal. Okay. Now, plants that have inversions or translocations can be reproductively isolated from normal plants. Okay, and that may be not such a bad thing because it can lead to new species. Okay, so cultivated rye and 4 wild species that are related to rye. Okay, translocations actually lead to the wild species being reproductively isolated from the cultivated species. Okay, so genetic exchange between cultivated rye and the 4 wild species are limited. Okay, and they have 14 chromosomes each, but still they still have limited contact with each other and limited crossing with each other due to the fact of the translocations. Okay, so here's a map of a chromosome, just showing you that corn geneticists as well as wheat geneticists, I think this is wheat chromosome, okay, actually have all these genes along them. This is just showing chromosome mapping, which scientists do, cytogenetics do, cytogeneticists do. Okay. Now, changes in chromosome number. Okay, so plants are remarkably tolerant of changes in chromosome number, as we saw last time. Remember those triploids and the tetraploids and the octoploids and etc. that we talked about. Okay, so let's see. So an aneuploid. So if an aneuploid has 1 or more chromosomes more, or 1 or more chromosomes less than a normal. Okay, so if one of these gametes is involved in fertilization, it can actually lead to cells, okay, that actually are aneuploids. Okay, that's what they're called. right? So, for example, cells in carrot have 9 pairs of homologs or 18 chromosomes. Okay, an aneuploid of carrot would either have 17 or 19 chromosomes. Okay, so aneuploids usually look different from their normal counterparts because of the missing or extra genes. They're just different looking. Okay, and if one of the gametes is involved in fertilization, it can lead to 2N gametes. Okay, so these 2N gametes, so sometimes meiosis fails to reduce chromosome number by half. That leads to gametes involving fertilization being 2N gametes. Okay, so they'll have at least one extra set of chromosomes, and that leads to what we call polyploidy, which we talked about last time. Okay, so polyploids oftentimes are larger, like sometimes they'll have larger flowers, or they'll have more flowers, higher yielding, or they'll have larger fruits, or they'll have more fruits than the other plants do, the regular diploids. Okay, so some kind of very famous polyploids include potato, which is a tetraploid. Okay, cotton, peanut, wheat, which are tetraploids and hexaploids. Okay, oats, strawberry, which is an octaploid, sugarcane, marigold, and lily. Okay, so marigold and lily, polyploids tend to have larger flowers than their diploid counterparts. Okay. Now remember that triploids tend to be sterile. They have no seeds, so seedless watermelon, also bananas are triploids. Okay, that's why those little black dots in the bananas, there's no seeds, right? In what are cultivated bananas. Okay. Now we're going to turn to Mendelian genetics. Okay, that genetics actually originated about 20 years before its significance was known. Okay, and genetics was actually, the father of genetics is Gregor Mendel. Okay, so he was an Augustinian monk who taught in the 1800s in Brno, in what is now the Czech Republic. Okay, so he actually did a wide range of studies in physics, mathematics, natural history, and in 1866 he published a paper on pea plants. Okay, which was very significant, but people didn't really understand his work. They overlooked his work, and he actually died in obscurity. He was a monk, so I guess he was taken care of by the church, but his work was ignored until 1900. Okay, when there were 3 botanists that reached the same conclusions as Mendel did. So each of these 3 botanists came across Mendel's paper by doing library research. Okay. Now the peas that he worked with are self-fertile. What that means is that the pollen from the anther of a flower can actually pollinate the stigma of the same flower, and the sperm from that flower can actually fertilize eggs in the ovary of the same flower. So they do self-fertilization because pea flowers are usually closed like this, so the anthers just normally fold down into the stigma. Okay. So generally pea plants are self-fertile. They do selfing. That's actually a horticultural term when you actually put the pollen from one flower on the stigma of the same flower. Okay. Okay, so peas are self-fertile. Okay, and what he noticed, okay, was that tall plants, after selfing for who knows how many generations, had tall offspring. Okay, so he wondered what would happen if he crossed a tall plant and a dwarf plant. Okay, so that would be the pea generation or the parental generation. He called it the pea generation or parental generation. Okay, so this is what you call a monohybrid cross. They actually have differences in one trait between the 2 parents. They're the same species, okay, but one was tall and one was dwarf. Okay, so what would happen if he crossed a tall and a dwarf? Would the offspring be intermediate in size? What do you think? Okay, think about it for a minute. So the F1, or the next generation, the first filial generation, filial means son or daughter, okay, were all tall, okay? And he didn't stop there. So the tall dominated over the dwarf, okay? So the F1 was all tall. So what he did is when he allowed the F1 to self, or if he sibbed it, or did a sibling cross of sister plants, okay, the second filial generation, or the F2 generation ratio, had 3 tall and 1 dwarf. Okay, there was a ratio of 3 tall to every dwarf that came about. Okay, so this parental trait in the P generation showed up in the second generation, even though it was masked or dominated in the first generation. So this is what the dwarf would be, what you would call the recessive trait, and the tall would be what you would call the dominant trait in Mendelian genetics. Okay, so you notice this ratio of 3 tall to 1 dwarf. This is important. He was the first to use quantified data. That means he actually counted the results of his crosses. He was the first to actually do that. Okay, so it was probably due to a lot of his work in mathematics. Okay, so he called the element that controlled the traits a factor. Okay, and he mathematically figured out that there were 2 factors for each trait, okay, such as tall and dwarf. Okay, so that's what we call alleles of a gene. Okay, we call them alleles. So a gene actually codes for a trait, and alleles are variants of this gene. Okay, alternate forms. All right? So these alternate forms of genes, or alleles, are located at the same location on homologous chromosomes. Okay? So in terms of Mendelian genetics, you have dominant traits and recessive traits. Okay? So the dominant traits mask the recessive traits in the F1. Okay? Now, the genotype is the genetic makeup of an organism. Okay? So usually symbolized by letters. So in the case of tall and dwarf, because tall was dominant, you name the genotype after the dominant trait, tall. That's why, so TT, capital T, was actually one parental type, okay, or the purebred for tall, okay. Little t was the other parental type, okay, or purebred for dwarf, okay. Now, big T, little t were the result of the cross between this, the homozygous dominant. Homozygous mean that they have the same alleles. Both alleles are the same. And the homozygous recessive. This is a recessive trait that actually has the 2 alleles being the same. And here you have a heterozygote with one big T, one little t. Okay? So if you cross 2 plants, okay, that have the same genetic makeup, or actually sib the 2 plants, you will actually have a different assortment coming up. You'll have 1 homozygous dominant to 2 heterozygous to 1 homozygous recessive. Okay? Now, so you have a 3 to 1 phenotypic ratio. You have a 1 to 2 to 1 genotypic ratio, or the genotypes. 1 homozygous dominant to 2 heterozygous to 1 homozygous recessive. Okay? But you'll have 3 tall to 1 dwarf in terms of the phenotypic ratio. Okay, we'll see that. So the phenotype is the physical expression of the genotype, is what the plant looks like, okay, or some other trait that is expressed. Okay, the genotype actually is stands for the letters stand for the genotype, and the phenotype is actually a descriptor, usually some kind of adjective. Okay, so tall and dwarf are the phenotypes, okay, of the plant involved. Okay, now homozygous, it means that the alleles are the same. would be capital T, or big T, or little t, little t. Okay, so these are purebreds. So the alleles are the same in these. Okay, heterozygous means that the alleles are different. Okay, so you have one big T, one little t for the heterozygous individual. Okay, so this is what they call a Punnett square. Okay, make a Punnett square, and you can put the gametes. So when the individual capital T, lowercase t, or big T, little t has meiosis happening, okay, the gametes can only are going to be half big T, half are going to be little t. So you put one set of gametes along one end. You put another set of gametes on the another end here. You bring this one down, this one over, because by convention you put the capital letter first. This one down, this one down, this one over, and this one over, and this one down. It gives you a 1 to 2 to 1 genotypic ratio, but you get a 3 to 1 phenotypic ratio. Okay? Now Mendel actually studied the pea plants, and the characters are as follows. Okay? So you had yellow seeds versus green seeds, with yellow seeds being dominant. You had smooth and wrinkled seeds, with smooth seeds being dominant. You had green and yellow pods, with the green pods being dominant. You had long and short stem, with the long stem being dominant, or tall being dominant. You had axial versus terminal inflorescences, with the axial inflorescences being dominant. And you had inflated and constricted pods, with the inflated pods being dominant. And you also had purple and white flowers, with purple being dominant. Okay? So all the dominant characteristics are actually listed here first. Okay? So we're going to do a monohybrid cross where we study a single gene. Okay? And so you are going to be assigned a genetics problem similar to the problem that we're going to do next. Okay? So we're going to have a genetics problem that's going to be your learning log 2 that will be due 1 week after this broadcast today. Okay? So genetics problem, so I'll show you how to do the genetics problem, and I will post the genetics problem on La Lima here under resources, okay, for you to do. And if you have any questions, of course, you can contact me, okay? So generally, you have the true breeding pea generation differ from one another, and they produce an F1, okay? Then the F1 is intercrossed and produces the F2 generation, okay? So doing a little bit of a different genetics problem, we might have a homozygous green podded crossed with a homozygous yellow podded individual, okay? When you do that, all their offspring have green pods, okay? So this is how you would set up the Punnett square, okay? So because all the offspring of this monohybrid cross in the F1 have green pods, you know that green are dominant over yellow, okay? So then you assign letters for the parents, capital G for the green individual, lowercase g for the yellow individual, because remember, you name the alleles after the dominant trait, okay? So you have big G for green, crossed with little g for yellow. And all their offspring will be big G, little g, okay? And all offspring will be heterozygous. And all the offspring will be green. So you know that green is dominant, okay? So the F1 is then crossed or selfed, okay? It would be big G, little g, crossed with big G, little g, okay? So if this one undergoes meiosis, the only possible gametes it can have in terms of its allele is either big G or little g. So about half will be big G, half will be little g. Same with this individual. Okay, so you could either cross it with another sister plant or you can self it. Okay, so here's the Punnett square. Big G, little g for one parent, big G, little g for the other parent. Okay, so you bring big G down, big G over for big G, big G. Okay, big G down, little g over for big G, little g. Big G over, little g down for big G, little g. And then little g down and little g over for little g, little g. Okay, so you'll have 1 big G to 2 big G, little g to 1 little g, little g as the genotypic ratio. Okay, you'll have 1, 2, 3 green potted individuals for every yellow potted individual. Okay, so that would be the phenotypic ratio. Okay, so there you go. This is the way I want you to present your answer, you know, with the genotypes actually written out along with the ratio. 1 big G to 2 big G, little g to 1 little g, little g. And then this is how I want you to write out the phenotypic ratio. Okay, 3 of the physical characteristic to 1 of the other physical characteristic. In this case, it's 3 green potted to 1 yellow potted individual. Okay? Anyway, that was quite something, right? So we'll go into the dihybrid cross and other crosses next time we come together. Okay, next time we have a lecture. Okay? I hope you enjoy this lecture. It's kind of fun to do a problem, right? So remember that your genetics problem will be posted up on La Lima, and it will be due 1 week from today, from this broadcast, okay? So we'll see you next time with a continuation of genetics and other subjects, okay? Aloha.